Hello students, we are discussing human health and diseases. In this chapter, in the previous class we studied the concepts like about health, about diseases and immune system, immunity. When come to the immunity, there are two types of immunity. One is innate immunity is also called as inborn immunity or non-specific immunity. So, in previous class, we discussed the innate immunity as well as barriers of innate immunity. Now, in this class, we are going to discuss a second type of immunity called acquired immunity. Acquired immunity is also called as specific immunity or adaptive immunity. Acquired immunity, in this type we are going to study in this class. One is definition, next unique features, then antigen, antibody, structure of antibody. Again, in this immunity we are going to study a two type of immunity called humoral immunity and cellular immunity. Okay, then when come to the definition, the body defines acquired by an individual during its life period against the specific pathogen due to previous infection is called acquired immunity. Acquired immunity is a kind of resistance to specific antigen or specific pathogen. The acquired immunity able to fight or produce the response to a specific type of pathogen. So it is also called as specific immunity. When come to the innate immunity, it is a non-specific. The innate immunity produces or fight against the all type of pathogen. There is no any specific one. It able to fight all kinds of pathogen like bacteria, viruses, fungus, protozoans, etc. But acquired immunity is specific fight to a specific type of pathogen, either virus or bacteria or protozoan. It is also called as adaptive immunity because the cells which take part in the immune mechanism or the lymphocytes and macrophages. So it is also known as a adaptive immunity. The lymphocytes and macrophages are able to take part in the immune mechanism, so that it is called as adaptive immunity. The acquired immunity able to adopt the person after the birth, the person able to acquire, receive, adopt the a type of immunity that is called as a acquired immunity. So the person able to receive from others, okay, receive from others or received from other sources, that is called as a acquired or adaptive immunity. This immunity developed after birth when encountered with pathogen. It supplements the immunity provided by the innate immunity. In the acquired immunity, there are two types of responses we are going to see. One is called as a primary response, another called as a secondary immune response. The primary immune response, it is the first response produced in the body during its first encounter with a pathogen. It takes long time to develop and lasts for a short period. Then when we come to the secondary immune response, the immune response produced in the body during its second encounter with the same pathogen. It is called secondary immune response. It is highly intensified and quick response, but it lasts for a long period. This is about the primary and secondary immune response. When come to the unique features of acquired immunity. First one, when come to the first one, the specific specificity, diversity, discrimination between self and non-self. Then fourth one called as a memory. So when come to the first a specificity. So the acquired immunity distinguish specific foreign molecule or specific type of pathogen. It fight the or produce the response specifically a specific type of pathogen. Then diversity. It recognizes vast variety of foreign molecule. The acquired immunity it able to recognize variety of a foreign or antigen or pathogens. Then third one, discrimination between self and non-self. It means it is able to recognize and respond to molecule that foreign or non-self. It will not respond to our own cell or molecules. The acquired immunity discriminate which are the body cells, which are the foreign molecule. So it protect the body cells, it fight the a foreign molecule. So it able to recognize then discriminate. Then fourth one, memory. After responding to the foreign microbes, this immune system retains the memory of that encounter. Then the second encounter with the same molecule occur. So the next secondary immune responses will be able to taken place. It means the acquired immunity having ability, there are some cells are there in the acquired immunity, those cells are able to memorize the a specific type of pathogen in the first encounter. The same type of pathogen comes in the body, so it able to memorize the a same type of cells or same type of immune response or same type of a fight. That is called as a memory. Then when come to the next antigen. So antigens are the foreign proteins that induce the immune system to produce the antibody. This is a foreign protein, foreign molecule. The antigens are located on membranes of microbes or cells. The antigen molecule has small regions or units. Those small regions or units are called epitope. It is also called as antigenic determinant. Okay. Then the epitope vary in their shape and size with specific antibody. Epitope, it is a region where the antibody able to react. Then one more term is there, immunogenicity. When the antigens enter the body, they induce the formation of specific antibodies by the lymphocyte. This is called immunogenicity. One more word is there, antigen reactivity. It means the chemical reaction between antigen and antibody. to form antigen antibody complex. This complex is called antigen reactivity. Then one more word, immunogen. Immunogen. So antigen which has both immunogenicity and antigenic reactivity, such type of antigen is called complete antigen or immunogen. So what the immunogenicity or antigenic reactivity are there? These two are the characters of antigen. If the antigen having a one character, then it is called as a, if antigen having a two character like immunogenicity and antigen reactivity, then it is called as a complete antigen or immunogen. Or if the antigen have antigenic reactivity but not immunogenicity, only antigenic reactivity is present but there is no immunogenicity, such antigen is called partial antigen or hapten. So this is about the antigen. When come to the antibodies are also called as immunoglobulins. Okay, immunoglobulin. It is also written as a Ig, capital I, small g, Ig. Antibodies. Antibodies defined as, or immunoglobulin defined as, these the special type of protein produced by lymphocytes of immune system in response to antigen. It is a special type of protein. Okay. So when antigen enter into the body, such antigen induces the immune system, then the immune system able to stimulate the B lymphocyte. The B lymphocyte able to indirectly produces a special type of protein to fight such type of antigen, that is called as a antibody. Okay, this when come to the structure of the antibody, the structure of immunoglobulin or antibody was discovered by two scientists. One is Gerald Morris Edelman. Second one, Rodney Robert Porter. Then the antibodies are produced by the immune system in response to the entry of antigen into the body. We know that all antibody molecules are immunoglobulins and chemically they are proteins. Okay, when it comes to the structure of antibody, the antibody molecule is made up of 4 polypeptide chain. 1, 2, 3, 4. 4 polypeptide chain. These 4 polypeptide chain arranged and look like a capital Y letter shaped structure. Then there are 2 long chains are there and 2 small chains are there. This is the small chain and this is the long chain. The long chain polypeptides or the long chain polypeptide chains are composed by 400 amino acids, like that two a short or small polypeptide chains are there, those are composed by 200 amino acids. Long chains are also called heavy chains. The short chains are also called light chains, and these are represented as a H2L2. All 4 chains are held together by disulfide bonds. These are the disulfide bonds. Then the polypeptide chains have two regions, namely variable region, another called a constant region. This is the variable region, this is called a constant region. This is the variable region, this is the constant region. Then the variable region is the antigen binding site. Antigen binding site that differ in different antibodies, where the constant region is a remain constant. So this is the variable region, this is the constant region. The constant regions remain constant, but the variable region are differ in different antibodies. In the variable region, there is a antigen binding sites are there. Those are we called as a paratope. In this region, the antigen able to bind. Okay, the paratope of the antibody, epitope of antigen. In that place, both are able to react with each other to form a antigen antibody complex. Okay, this is about the a structure of antibody or immunoglobulin. When it comes to the types of antibody, there are different types of antibodies are there. In that, the major one are first one IgA, IgD, IgE, IgG and IgM. IgA, this antibody is also called secretory immunoglobulin. It is present in the colostrum of mother milk. Colostrum means the first milk produced by a mother after giving birth to the young one. This IgA effective against the antigen. Second, IgD, immunoglobulin D, the smallest quantity antibody, it effective against the toxins. Then IgE, this antibody associated with the allergic reactions. Next, IgG, it is constitutes 75% of total antibody. It is a predominant antibody. Okay, it is a predominant antibody. It presents everywhere. Then IgM, it is a physically largest antibody. This IgM antibody very first reach the infection. It is the first antibody fight or defend against the antigen or infection. These are the types of antibody. When come to the next is difference between antigen and antibody. The antigen is also called as immunogens. Antibody is also called immunoglobulins. Antigen stimulates the production of antibody. Antibody is produced in response to antigen. Then antigen may be protein or polysaccharide or lipid. The antibody is a special type of protein molecule. Antigen is a variable in structure, but antibody is a capital Y letter shaped structure. Then antigen is a foreign substance bring out reaction in the body. The antibody is a protective chemical produced in the body. This is about the difference. When come to the next concept is Acquired immunity carried out special type of lymphocyte cells. Okay, lymphocyte cells. Before then, the lymphocytes are very much play role in acquired immunity. From those lymphocytes or on this basis of lymphocytes, the acquired immunity is also divided or classified into two types: humoral immunity and another called as cellular immunity. Yes. When come to the first, lymphocytes, these are the WBC cells, originate from the bone marrow, formed in the bone marrow. So, how the lymphocytes are formed in the bone marrow? The 50% of the lymphocytes are reached to the thymus. In the thymus gland, they are differentiate into T lymphocytes. Those we are called as T cells. The remaining 50% of lymphocytes are do not reach the thymus. They are differentiated in bone marrow itself. So these lymphocytes we call as a B lymphocytes. B lymphocytes. In addition to B and T lymphocytes, there is another type of lymphocytes are the natural killer cells. We already studied. Then, when come to the two type of immunity, one is humoral immunity, is also called as a antibody mediated immunity. Antibody mediated immunity. Then, when come to the antibody mediated immunity or a humoral immunity or role of B lymphocytes. B lymphocytes. When come to the first one, definition of the humoral immunity. The B lymphocytes acts indirectly against the antigens by producing antibody. This is called humoral immunity, or also defined as the immunity which is the immunity in which the body fight against the pathogen by producing antibodies into body fluids is called humoral or antibody mediated immunity. The immunity which produces the antibodies is called humoral immunity. Okay. What the a humoral immunity produces the antibodies, nothing but the B lymphocyte produces the antibodies. Those antibodies are carried by body fluids like blood, lymph. Okay, then those antibodies are moved in different parts of the body. Yes, when the antigen enter into the tissue or enter into the body, the B lymphocyte cells are stimulated to produce the antibodies. First, there are thousands of antigens enter into the antigen enter into the body. Then the B lymphocytes are able to active or stimulated. Those B lymphocytes are stimulate the plasma cells. Plasma or activate the plasma cells. Then the plasma cells are able to produce the antibodies. So one thing is clear, the B lymphocytes are not directly produces the antibodies. The B lymphocytes first they are get the stimulation when the antigen enter into the body. After that, the B lymphocyte activates or stimulate the plasma cells. Then the plasma cells produces the antibodies. Then the antibodies are multiply its number. Those nearly the rate of antibody multiplication is 200 to 2000 molecules per second. Then those antibodies are combined with the antigen to form antigen antibody complex. Then the reaction is taken place. Okay, this is about the humoral immunity. When it comes to the cell mediated immunity, are also called cellular immunity or role of T lymphocytes. The T lymphocytes, sorry, the when come to the definition, the immunity in which the body fight against the pathogen by producing special type of cells like macrophages. Such type of immunity is called cell-mediated immunity. In humoral immunity, antibody production. In the cellular immunity, the cells are able to produce. Okay. Then it is also defined as the T lymphocyte acts directly against the antigen by special type of antigen. The special type of cells is called cellular immunity. There are about thousands different types of T lymphocyte cells. T lymphocyte cells are able to produce against the specific antigen. Okay. When the antigen enter into the body, then the T lymphocytes are able to produce a number of different types of T lymphocyte cells. The different type of T lymphocyte perform the different type of function. The different type of T lymphocytes are majorly one killer T cells, helper T cells, suppressor T cells, memory T cells. When come to the killer T cells, these cells migrate to the site of infection and attach attach to invading the microbes or antigen and secrete a toxic substance like cytotoxins. Those cytotoxins destroy the microbes directly. This is the function of killer T cells. This is the first cell able to fight against the infectious pathogen or particle. Second, helper T cells. These cells stimulate the B lymphocyte cells to produce the antibodies which interact with the antigen of the microbes. If the killer T cells fails to fight the antigen, then the helper T cells are able to produce. Those helper T cells are able to stimulate the B lymphocytes. Then the B lymphocyte produces stimulate the plasma cells. Then the plasma cells produces the antibodies. Then the antibodies plus killer T cells both are able to fight the antigen. Then third one, suppressor T cells. These cells suppress the immune system after the antigen or microbe is destroyed. If the killer T cells and antibodies of helper T cells both are able to completely fight the antigen, then what happens? The suppressor T cells able to suppress the immune system, suppress the production and function of the killer and helper T cells. Then fourth one, memory T cells. These cells are pre-programmed to recognize and respond very quickly against an antigen that may appear in the future. So what the killer T cells, what the helper T cells perform their function against the specific type of antigen, all the functions are able to memorize the memory cells. Okay, memorize the or recognize the complete memory cells. If the same type of pathogen occurs in future, in second time, then what happens? The memory T cells are able to memorize, stimulate directly to the killer T cells to kill the specific type of pathogen. Okay, this is about the a cell-mediated immunity. When come to the difference between the B lymphocytes and T lymphocytes, the B lymphocytes are produced in the bone marrow and differentiated in bone marrow itself. The T lymphocytes are produced in the bone marrow but differentiated in the thymus gland. Next, the B lymphocytes, on exposed to the antigen, become active, multiply to produce the produce the plasma cells. Then the plasma cells secrete the antibodies. It means the B lymphocytes indirectly produce the antibodies by stimulate the activate the plasma cells. The T lymphocyte, on exposure of specific antigen, it become active and multiply and produce different type of T lymphocyte cells like killer T cells, helper T cells, suppressor T cells, and memory T cells. B lymphocyte cells attack antigen indirectly by producing the antibodies. The T lymphocytes directly attack the antigen. The B lymphocytes secrete the specific type of antibody. The T lymphocytes secrete the cytotoxins and other chemicals to destroy the pathogen. The B lymphocytes, they have short lifespan. The T lymphocytes have long lifespan. Long lifespan. Then the B lymphocyte do not migrate to the site of infection. The T lymphocytes migrate to the site of infection. Okay, this is about the acquired immunity. Then there is some other characters are there. The rejection of organ transplant like heart, kidney, eyes, liver are due to the lymphocytes. Based on the lymphocytes, the organ transplantation rejection is taken place. If heart or kidney size, shape, everything is matches for the transplantation, but the T lymphocytes will not able to match, then the rejection will be taken place. Then tissue matching, blood group matching are essential for organ transplantation. Not only the tissue matching, blood group matching, also the T lymphocytes also match. Even after tissue matching, immunosuppressant is required before and after transplantation. Okay. Then the immune system is able to differentiate self and non-self, and cell-mediated immune response are responsible for the organ rejection. The immune system differentiate the body cells and the foreign cells. That is the self and non-self. So when we are going to conduct the organ transplantation, so here not only require for the blood group matching, tissue matching or size, shape, it also we consider the T lymphocytes. It is also very important. If the T lymphocytes will not able to match, then what happens? Suppression of immune system taken place, the rejection is occur. So this is about the acquired immunity. Next we are move on to the next concept. Okay, thank you.